The first speaker today will be Lidia Morawska, Distinguished Professor of Earth and Atmospheric Sciences, University of Queensland, Australia. Professor Morawska will start us off by talking about the source side of the aerobiological pathway in a talk entitled, How Particles Are Generated During Our Respiratory Activities. Thank you.
Very good afternoon from sunny Krakow, where I arrived about an hour ago from London, worrying that my flight would arrive on time, but it did. Well, I, in a way, continue from what Dawn started, the issue of definitions. Is it aerosol or droplet? Well, I'm coming from aerosol science, so aerosol is an assembly of liquid and solid particle suspended in a gaseous medium long enough to enable observation and measurement. And droplet is a liquid particle and nothing to do with size. But as we know, in medicine, it's different. Aerosol are small particles, droplet larger particles. That's how Dawn explained, and the divisions between them. Well, we won't worry about this. And my approach, since I've realized these problems and the difficulties in kind of, deciding who would want to give up which definitions, I'd say let's call them particles. And this way it's properly from both areas of the disciplines, and we don't worry about this. So I will call it a particle or particles. And we are a source of respiratory particles. We all have respiratory fluids, as we know. Particles are formed from the fluids during all our respiratory activities, and they are emitted through the mouth or nose. Well, sometimes we can see them, like with this salivating dog, but we can also occasionally see them when talking to an overexcited colleague. So how are they aerosolized? How are they formed? If we start from the process of aerosolization, the definition is such: it results from the passage of an airstream of a sufficiently high speed over the surface of a liquid. Now, where is this airstream and where is the liquid? It is in different parts of our respiratory tract. From a very simplistic point of view, we can compare aerosolization to what happens in this old-fashioned perfume bottle or a nebulizer, which many of us use for many different purposes. Now, the one part of the complexity is that our respiratory tract contains not one, but many nebulizers. And not only. The aerosolization is not the only process in which particles are generated in the respiratory tract. There are several different other processes. In particular, if we look what happened in the deepest part of the respiratory tract, in the bronchiole. Now, it's not aerosolization, but it is the burst of particles during inhalation and production of the particles. So that's that a narrowing passage and then bursting when it opens. Slightly different process takes care in larynx. This is aerosolation during voicing, during vocal cord vibration. And then in the mouth, there are several processes, but here we've got saliva and aerosolization and other processes between the tongue, teeth, and lip during speech articulation. So as you can see, there's quite a complexity into this, and this is not the end of the complexity. So the particles are formed, and before they are emitted, they already undergo various processes. And in particular, some of them deposit already before they are emitted, which means this changes the initial size distribution. And then there was this fascinating paper published in Science 2 years ago about this viscous bubbles and what happens. And these things, it was pointed out by the authors, can happen in the respiratory tract, leading to a special type of particles. So it is complex. just showing this in a cross section, this process in the bronchiole region. If we start from the top and air flows towards the right, so the bronchiole is fully open, and then as air continues flowing, it contracts, and eventually it's contracted completely and filled with liquid. And then air starts where we are breathing in and out, air flows in the opposite direction, and here the bubble sort of pops. So this is generation of the particles. Well, it's all good, but we cannot measure this process directly, but model and simulate. Maybe sometime in the future, we will have some kind of nanobots or whatever, which we could send down the respiratory tract, but this is not possible. And any attempt, well, there have been many attempts of doing this based on some experimental setups and so on. Well, it helps, but it is not exactly what's happening. So the level of understanding is good qualitative, but very limited quantitative, what exactly happens there. So now, detection of these particles, detection after they leave our nose and mouth. I'll say a few words about instrumental techniques. I remember at the beginning of the pandemic, many of my colleagues, particularly medical colleagues, would say, well, bring your particle counter and do measure this or that. Well, it's normally not that simple to just bring a particle counter and measure something. If you want to measure something meaningful, it's much more complex. This is a setup of our initial experimental flow tunnel, in which we measured size distribution and concentration of particles emitted during different respiratory activities. So as you can see, the particles are flowing down with this straightened flow. And then down the track, we had several different types of instruments for measuring particles of different sizes, because as I'll explain in a minute, there is a great variation in sizes. That's how the tunnel looks from inside and from the side. But there are other instrumental techniques. For example, this one, a study conducted by Zayas and colleagues. This is, of course, aerosol on an open bench. And here they use a different instrumentation altogether, laser diffractionometry system, an open bench. What they measured was different, and this gave a different kind of angle to our understanding. None of this, whether the first set which I saw or the Zaya set, wouldn't allow us to deal with the issues of low concentrations, or if we want to keep the particles for any kind of analysis. So in our latest study, we designed this rotating drum, and here the particles are accumulated. And this was very useful because, first of all, we could achieve higher concentrations, which we couldn't in a flow tunnel. Flow tunnel gave us exact age of the particles, but concentrations were low. Or we could test how long pathogens stay viable in infectious in a system like this. But it's not suitable for fast changing processes. So there are many, many different experimental approaches, and it all depends what's the aim of our study. So size distribution of particles. This is what came from some of the experiments with our original flow tunnel. So it's a bit complex diagram, but hope I'll be able to explain exactly what's happening here. So on the horizontal axis, logarithmic scale, we've got particle diameter and particle concentration also on logarithmic scale on the vertical axis. So first of all, we can see that the majority of the particles, the peak is in this smaller size range, and there's significantly higher majority because of the logarithmic scale. So the particles are basically 10 micrometers and less. The importance of this is that these particles are light and small enough they can stay suspended for quite a long time. You can also see these different modes here. The first one is bronchiole fluid film burst mode from the deepest part. And it's believed that this is where H5N1 virus originates and resides. Slightly bigger mode is laryngeal vibration mode, and the biggest one, oral speed articulation, the biggest and the widest. And this is where the source of H1N1. Now, something to point out that this looks nice, small, smooth curve, but it's not something which comes just from one instrument. It required 10 data processing steps to get to that point. And I'm also pointing out to the scale. We are talking about particle number concentration. So the level of understanding of this is good, quantitative, and replicated in numerous studies, other studies than ours as well. Now, where's the virus in these particles? Because those particles were just any particles, and the particles, the physics of particles don't really care whether there's virus or not. Virus is this virus, SARS-CoV-2, naked virus, is about 0.12 micrometer, which immediately tells us that the size of virus-laden particles has to be larger than this, because particles, when they are formed, aerosolized, or formed in any other of these processes, they contain water, mucus, salts, and so on, so they are bigger. How much bigger? Well, it's been demonstrated that particles smaller than 1 micrometer in general contain higher loads of SARS-CoV-2. Not that one individual particles contain such a high load, but because the majority, the vast majority are in this size range, that's where the highest load is. So level of understanding, a reasonable majority with mid micrometer to early micrometer range. Now, this is also along these lines in terms of where the virus is, obtained from very different type of studies, measurements in mass size distribution in mixed rooms, hospital rooms. Now, this was done by two types of instruments. So first of all, aerodynamic particle size are similar to what we measured, which gave the researchers the size distribution. But also they used NIR sample to collect for biological sampling. And since this is in the rooms, so there are all respiratory activities and the background. Now, I'd also like to point out that there were several data processing steps, so it's not just coming from the instrument. And also, please note the scale, because you may say, okay, what I saw before, the majority was in the submicrometer range, and there was little in the larger size, and here's the other way around. Now, this is mass distribution. The point is that the mass of a few larger particles significantly overshadows many, many more small particles, but this is just mass. But the most important point which I wanted to show here is that the highest load was in this smaller size below 1 micrometer. So this was a study by Sandarpia and colleagues. So concentration of the particles in the air after we understand their size distribution. Concentration sometimes expresses concentrations, sometimes as emission rates. Now, again, this is from the first study which I mentioned. So we've got the activity, and the activities including breathing, nose, mouth, counting, voice, whisper, and then concentration on the vertical axis. So first of all, we can see that all respiratory activities contribute to particles, but breathing the least. Well, on the other hand, we breathe all the time. What contributed the highest concentration was singing. It wasn't quite singing, it was this that what we were able to reproduce in this study. Now, there were other similar studies much later conducted, well, sometime later, 10 years later, study by Asadi and colleagues, and they showed very similar results. with different activities from nose to mouth breathing and then loud speaking. And again, as you can see, breathing the least and speaking, loud speaking, the highest concentrations. So what we can say that there's a good quantitative understanding of relative concentrations emissions. Now, there are differences due to different instrument systems used. Now, one can say, well, that's a problem if we have these differences, but we can explain this very well if we wanted to bring them together to the same kind of scale, we could. But this is the reason that different instruments and systems are used. There's insufficient data for different age groups and susceptibilities. Now, just something which was published recently during this pandemic, more on how loudness affects particle emissions. I thought that was a very interesting study, and going into depths of this loudness with exactly measuring decibels of different activities. So you can see how this experimental setup was arranged with this funnel here, and then the air was going, I think it was to the APS, aerodynamic particle sizer, but in addition, this loudness was measured. Now, happy birthday. So here we've got the different activities on the horizontal scale and then normalized particle mass concentration. What I wanted to show and compare is this one. The top here is the loudest happy birthday. And we compare this with breathing out of mouth. So, well, first glance say, okay, happy birthday is more than breathing. But if you look at the scale, it's logarithmic scale, vertical scale. So we are talking almost 100 times more when we are singing than when we are just breathing. That's something to keep in mind. And there's also something interesting, a study to which I was invited on this topic of the impact of airway hygiene. What's airway hygiene? Now, airway hygiene is to make sure that airways are hydrated. There's always a problem when airways are dry. So this hydration here we're using to drug-free nasal saline formulation and then comprised with this salts. It was conducted in 3 different locations across the United States and India in Bangalore. And well, these are the details how it was measured. The measurement itself was relatively simple, again with a funnel and going to a particle counter. Now, so what was the outcome of this? That was the study, first study published on this topic. I highlighted the outcome, but I emphasize it here and stress. So respiratory droplets, what was measured, diminished in number on exhalation by up to 99% via the airway hygiene administration of nasal saline rich in calcium. That was quite amazing. We just couldn't believe this, that the impact was so high. And I must say that first, without sort of thinking much about this, I thought, well, there will be more liquid, perhaps there will be more particles. There's no clear explanation yet. Is this because of the changes to lung surface characteristics, flow pattern, something else? But it sure shows the significance of this aspect of hydration of airways. Now I'm stressing the factor of just exhalation, but this was the initial objective of the study, was to look on the impact of deposition of particles from polluted air. So with this, thank you. I won't summarize this again because I tried to summarize every aspect of this. So I will be looking forward to answering your questions later. Thank you.